All right, hello and welcome back. We are going to continue to look at internal and external structures of plants, and we are looking at leaves and seeds in this lesson. The main function of leaves, as you should be aware of, is to trap sunlight. Leaves may be modified, meaning that the shape might be different than what a typical leaf would look like, and they may be modified to store food. An example of a leaf that stores food would be onion. Leaves have stomata. Now, stomata is a plural of the word stoma. So each pore that the leaf contains, you call that pore a stoma. And the collection of those stoma, or the plural of stoma, would be stomata. So the purpose of the stomata is to allow the exchange of gases. And you usually find the stomata on the lower section of the leaf. This diagram is showing the external structures of a typical dicotyledonous leaf. So we will quickly identify the structures that I mentioned here. So at the top, the very top, we call that the apex. Here it is labeled as a tip, but the correct word would be the apex, A-P-E-X. Then at the middle, we call that the midrib. And this is where the main vein runs through the leaf, and in that, it contains the xylem and the phloem vessels. Then we have the margin, and this is referring to the edge of the leaf. We have a network of veins, and the network of veins branches off from the midrib. The veins contain xylem and phloem tubes. Petiole holds the leaf onto the stem, and then we have the lamina. The lamina would be the flat structure of the leaf, or the flat surface of the leaf. Here we have the internal structures of the leaf. This is when you cut the leaf, you take a section of the leaf and you place it under a microscope, a very powerful microscope. Of course, what you would see is not as neat as you are seeing currently, but this is the animated drawing of the internal structure of a leaf. So the structures, we have a cuticle, which is not really a structure, it's really a layer at the top of the leaf. It's a waxy layer, and the upper epidermis is what is secreting this cuticle. And we will look through that, the function of the different parts shortly. Then we have the upper epidermis and the lower epidermis. We have the palisade layer. So the palisade mesophyll and the spongy mesophyll, together we call the two layers mesophyll layer. So the mesophyll layer is divided into the palisade mesophyll layer and the spongy mesophyll layer. We have the stoma, which is the pore. And surrounding the stoma, we have guard cells. They're like guards that allow the opening and the closing of the stoma, or they're like gates that opens and close. And we have the vascular bundle. The vascular bundle consists of the xylem and the phloem tubes. So as I mentioned, we will go through the structures one by one and look at the function of each one of the structures. So we're looking here at the cuticle. So the cuticle is a waxy layer at the top, and the purpose of it being waxy is actually to prevent excessive water loss. Think of putting a layer of oil over water. The layer of oil over the water will prevent things like oxygen and so forth from entering, so it creates a barrier. So similarly, the cuticle, the waxy layer, is creating a barrier and prevent the evaporation of water. Next, we have the epidermis. As I mentioned, we have the upper epidermis and we have the lower epidermis. The lower epidermis, it contains stomata, which allows the movement of gases, as mentioned before, and they are surrounded by two guard cells. In this slide, we're looking at the mesophyll layer. So as I mentioned before as well, we have the palisade mesophyll layer. And the palisade mesophyll layer, this is a layer that contains a lot of chloroplasts. In other words, here is where most of the photosynthesis is taking place. And it would make sense that the palisade layer is at the upper level because the sunlight will reach the palisade layer very easily. and that will allow photosynthesis to occur very quickly. So the cells in the palisade layer, they are usually very closely packed. This diagram shows a little space, but in a real situation, in a real plant, you would not have so much of space in the palisade layer. The cells are closely packed, they're touching one another without having any space between them. And then we have the spongy mesophyll layer, labeled number 4 on the diagram. And the cells are loosely packed, meaning that there are spaces, and the space is to allow the movement of gases and also water vapor, which is in a gas state. There are fewer chloroplasts in a spongy layer, and there's one of the reasons why is because it's at a lower section, so sunlight will not penetrate so deep down into the leaf. Very small amount of sunlight reaches this location, so not as much photosynthesis will be taking place in the spongy mesophyll layer. Next, we have the vascular tissues. They are present in the veins of the leaves, as mentioned before, and we have two of them. We have the xylem and the phloem. So the xylem, main function of the xylem is to transport water and transport the dissolved minerals. So there are minerals such as magnesium, might be calcium and so forth. These are dissolved in water. So as the plant takes up the water, it is also taking up those minerals. The movement of the water is usually in one direction, meaning it moves from the root and it moves towards the leaves. The phloem, the purpose of the phloem tubes, they translocate. Translocate means they're moving it from one location to another. So they translocate manufactured foods from the source. And the source of the manufactured food will be the leaves, because that's where photosynthesis happens, to different parts of the plant. To different parts of the plant that needs it. The different parts of the plant may need it to store it. For example, some of the plants may store excess food in their stem, like sugarcane. So the food is translocated from the leaves to the stem, or it can be translocated to the roots, or wherever it is needed. Whenever you have drought, for example, well, then opposite may happen. The stored food, let's say it is in the roots, the stored food in the roots is translocated from the roots towards the leaves and other parts that it is needed. All right, so that's it for the internal and external structures of leaves. We're now going to look at the internal and external structures of seeds. So the seeds of dicots and monocots plants are quite different. And you can look at the image and see the difference right away. There are certain structures that are in the dicot seed that looks quite different from that of the monocot seed. But one of the major differences that you should be able to spot and identify is that the monocot seed, they have one cotyledon, and that is why it is called monocotyledon. Mono meaning one. So monocot have one cotyledon, and the dicot, di is referring, the prefix di, di, is referring to two. So the dicotyledonous plants, or the dicotyledonous seed, have two cotyledons. And what's the purpose of the cotyledon? The main purpose of the cotyledon is to store food, and the stored food will be used during germination. So quick look at the external structures of a dicot seed. We have the tester. You may also refer to it as a seed coat, but the better term to use would be the tester. The purpose of it is to just an outer coat, so it protects whatever is under it. And whatever is under it is the cotyledons and the plumule and radicle and so forth that are developing. Then we have the micropyle. The micropyle is a tiny pore, very small pore, that allows water to enter. And before germination can occur, water is very important. Water must enter into the seed before germination can occur. So the micropyle serves that purpose to allow the movement of water into the seed. Then we have a structure, and that's called the hilum, or a scar. And that scar is really showing where the seed was attached to the pod. For example, if it's a bean seed, it was attached to a pod, or the ovary wall for the other types of seeds. So internal structure, we're looking at, if you remove the tester, how the seed will look below. So you will see the cotyledons. Also, sometime before it was the seed leaves, as the plant germinate, you will see that these two will be showing above the ground, and these contain the stored food. Under the tester, the internal structure, you should also be able to see the plumule, which is the young shoot that is developing. Then we have the radicle. All right, so the monocot seed, the outer protective coat is formed from the fusion of the tester and the fruit wall, as the maize grain is a one-seeded fruit. So if you look at the structure of the maize, that is the corn, you will notice that the outer protective structure is fused with the tester. So you have a fusion of the fruit with the tester. There's only one cotyledon present, and they have another food storage structure called the endosperm, and which is separate from the cotyledon. All right, so that brings us to the end of this lesson, where we looked at the internal structure of the leaf and seed, both monocotyledonous seed and dicotyledonous seed. So as always, if you have any question regarding what we discussed in this lesson, please do not hesitate to reach out and ask those questions that you have on your mind. All right, until the next lesson, I wish you all the best.